We have a crisis at the moment. We're rapidly running out of antibiotics which are effective. Drug discovery is hugely expensive. Current estimates are anything between $2 to $3 billion to get a molecule all the way through into the marketplace.
I'm Gavin Freeborn, and welcome back to Original Ideas, a podcast from the University of Liverpool. In this series, we explore the world of academic research, the impact it has on society, and how it can shape our future. We also look at the latest research and meet the people behind it. In this episode, we're exploring a world that holds hope for so many: therapeutics innovation.
Our overarching aim here is to really improve drug development and the delivery pathway. So we at the university have the expertise and facilities that span that entire normal drug development pathway.
Researching and delivering effective medicines are essential for the prevention, treatment, and cure of diseases around the world. And there's an urgent need for the development of medicines that are both clinically effective and cost efficient. The team at the University of Liverpool have a rich history of groundbreaking discoveries and transforming research in infectious diseases and global health into real-world medical solutions. It's a really interesting topic that will impact every single one of us. So let's explore what the future holds for therapeutics innovation. I caught up with Professor Neil Liptrott, Surgeon Scientist Rajarshi, Mukherjee, and Dr Danielle Brain to find out more. An interesting conversation ahead as ever. Neil, Rajarshi, and Danielle, thanks for joining us on the podcast. So before we begin, could you give us a very brief overview of your background and work?
So my name is Rishi Mukherjee. I'm a surgeon scientist working with the University of Liverpool. I've been interested in problem solving as a medic for many years, and I have a passion for surgery and science. And I've been focused on the problem of acute pancreatitis, which many regard as an untreatable disease, which after 200 years of scientific research endeavour and over 200 negative randomised controlled trials still lacks any targeted drug treatment. So that voyage of discovery is something that I'm still on, and recently I gained a Clinician Scientist Award as part of the inaugural MRC Medicines Development Scheme, and I'm working with a major pharmaceutical company to develop new biomarkers and drugs to treat this potentially devastating condition.
Neil?
I'm Neil Liptrott. I'm a professor in the Pharmacology and Therapeutics department here at Liverpool. I'm an immunologist by background, and my group's research interests are aimed at complex medicines, advanced therapeutics and advanced materials, particularly how they interact with the immune system to either cause potentially adverse events or how we can use those materials to engineer immune responses that we actually want for therapeutic benefit.
Danielle, if you could give us a very brief overview of your background and work.
So I'm Danielle Brain, and I'm a postdoc at the University of Liverpool, and I work within Neil's group here at Liverpool, and my research mainly focuses on the immunocompatibility, so the interaction of the immune system with novel therapeutic delivery systems. At the moment, my projects mostly work on LNPs, so a delivery system that was used in the COVID vaccine. So I look at that and also look at developing the models that we use to test these delivery systems.
Brilliant, thank you. Now, firstly, if we can start with you, Neil, can you please break down therapeutics innovation for us? What are we talking about when we discuss this?
So I think most people's view, and it's not necessarily incorrect, is that therapeutics innovation is creating new therapies for particular applications. So, really sort of focused on blue sky research and development, which is obviously a significant part of moving that frontier forward. But also it encompasses repurposing existing therapeutics in different ways. There's some really good examples of that where low molecular weight drugs have been used off license for different applications and diseases and they were originally designed for. So it's not just about creating new therapeutics and new molecules. It's about how we can potentially reuse or reformulate existing therapeutics, either for a different application or to improve things like the pharmacokinetics or the safety of the drugs that have been developed originally. And obviously, nanotechnology, nanoformulation is something, it sounds very modern, but actually it's been around for decades. And I think that that's one of the sort of the biggest advantages is with that kind of technology, that you can overlay that advancement in material science and formulation and so on and so forth to improve therapeutic indices of existing drugs that may be already slightly problematic for different reasons.
And can you share some recent innovations in drug delivery? What have you seen in your work?
So I think that probably the slightly obvious one, but I think it's worth highlighting and mentioning it because of the impact that it had globally, was the development of the mRNA LNP vaccines for COVID. So LNPs, lipidic nanoparticles, there are lots of different types of them. They've been around for a very long time, both sort of scientifically and clinically. So the first ever FDA-approved nano drug was a therapeutic called Doxil, which is still used to treat certain types of cancer. So we know that LNPs have been around for a long time. mRNA technology itself has been around, not as long as LNPs, but it's been around for a while. But obviously it was the first time that these things had kind of come together to combat a major global threat. They weren't the first nucleic acid LNPs to be approved. That was for a slightly different condition, but they were certainly the first ones to enter a huge scale-up and manufacture and deployment. And, I think that not only is the technology interesting, I think that it was a turning point in sort of the application of those kind of technologies to biomedicine and healthcare, that you can see what can happen when everyone is pointing in the same direction. From both a funding perspective and clinical trials, people think that these technologies kind of came out of nowhere. They didn't. They've been around for a while, but that huge both financial and logistical and human effort was necessary to actually get them into the clinic and, get them into people's arms effectively. So I think that that's been one of the biggest impacts for those kind of advances in technology. But we see, or as we go forward into biomaterials, I think that there's a lot more on the horizon that's coming. So it's a very exciting time, but also it means that there's a lot more things that we need to consider about when we're developing these therapeutics. Innovation is fantastic, but we need to make sure that we are moving everything forward at a reasonable pace.
Brilliant. Thank you very much. Rishi, if I can come to you, your background is quite interesting in that you moved from being a clinician to working in academia. What developments have you seen in your area of research that are helping shape drug discovery?
So, firstly, just to reframe the understanding of what I am as an individual, I think that the common perception is that clinical medicine and science are binary choices, black and white, and I'm of the breed of medic that I've chosen the third way, as it were. So ever since medical school, I've been interested in combining the two in terms of medicine and science, and pursued a career in academic surgery as a result. And I've particularly focused on pancreatitis because it's something that I realized very early on was a condition that had a huge unmet need. It can be a devastating condition with potentially months on ITU, and in the severe cases can lead to death. And I questioned why, as a medical student, we had to write it off compared to other conditions, saying we simply don't have any targeted treatments for this, so everything is just supportive. And through that process, the voyage of discovery I've been on and the developments I've seen in therapeutics innovation over the last 15 years or so, starting with my doctoral degree and going through my postdoctoral experience, have focused on three main areas: predominantly better biology, better models, and also better drugs. So if we consider the better biology, I've been looking at the way that mitochondria, a little organelle in the cell, which normally provide energy and are the powerhouses of the cells, can actually determine whether the cell dies or survives, and also lead to inflammation throughout the body. And there was a key protein that we found that if you inhibit or modulate, you can affect the severity of the disease process through that whole approach. So when it comes to models, we've developed new ways of testing our hypotheses through models that are more relevant to the human condition. There's been a great focus in the past on animal models, and we've developed innovative methods of using digital techniques to standardize assessment so that we can multiplex and develop methods that assess multiple factors simultaneously at the same time. We've developed large biobanks of materials that allow us to get a better idea of the actual relevance of certain discoveries to the human condition and translate findings accordingly. And then when it comes to drug development, which is probably the most exciting area, through collaborations with medicinal chemistry in the university and Professor Paul O'Neill and my own mentor, Professor Robert Sutton, the head of the Liverpool Pancreatitis Research Group, we've been able to develop novel small molecules that target cyclophilin D within the actual cells of the body and allow us to decrease the severity of pancreatitis in preclinical models. So we're very hopeful that we'll be able to take that forward to the next stage and progress on to phase 1 trials in humans, hopefully in the future.
Brilliant. Thanks very much. Danielle, as recently as last year, I know you were awarded funding for a particular type of research around developing a 3D skin model for patients who need injections into the skin. Could you tell us a little about that?
Yes, so last year I was awarded some funding for a researcher in residence scheme, and this scheme allows me to spend time at MDC, so the Medicines Discovery Catapult, and this is to help develop a model for testing immune interactions to therapeutic delivery systems that will be delivered subcutaneously. And so my project is to develop an in vitro, so a lab-based kind of assessment, and this is going to be a 3D subcutaneous kind of skin model, and this is to assess the immunocompatibility of novel therapeutic delivery systems. And so immunocompatibility is the degree to which a material or substance is compatible with the immune system, and this means that it doesn't trigger a strong or harmful immune response. And this is particularly important if this therapeutic is going to be administered in humans and doesn't cause a detrimental effect in humans. And so assays like this are vital for the development of new therapeutics and down-selecting therapeutic candidates. And this project is particularly really useful for me because it allows me to kind of develop my career as a scientist, and I'm undertaking my first ever project where I'm leading the project, and I'm also able to learn new skills at the Medicines Discovery Catapult, where I'm able to learn and leverage their expertise in microscopy in order to develop this model and to be able to kind of image the cells within this 3D model so that we can then use it to down select candidates and test these therapeutics.
Excellent. Brilliant. Thank you very much, Danielle. So a question for you all now. There seems to be so much work and research in therapeutics innovation. What one thing are you most excited about for the future, Rishi?
So undoubtedly, I have to say for myself, it's artificial intelligence, and where artificial intelligence and machine learning will take drug discovery and therapeutics innovation in the future. The promise is immense. Artificial intelligence could possibly, make more effective treatments that are more efficient in a more rapid time to a greater number, more diseases that have been neglected for some time. Firstly, if we take efficacy and improvements that AI can offer in terms of drug discovery and efficacy, I like to use the analogy of football. I am a Liverpool fan, and it's a little bit like our traditional approaches have been like having a free kick at 40 metres outside the box, which I think has a sort of success rate of about 40%. And it's similar to the sort of traditional approaches to drug discovery. Whereas the promise held by artificial intelligence is like Mo Salah getting a penalty within the box, targeted treatments that can deliver 90% in terms of efficacy at phase 1 trials. And that's borne out in reviews and studies that show that AI-driven drugs are much more effective in terms of their efficacy at phase 1 studies. Secondly, it's the efficiency that artificial intelligence offers. It's much better in terms of screening of a variety of compounds, multiplexing, and a recent landmark discovery published by a US and Chinese group in Nature Biotechnology showed that the whole process can be sped up by about 10 times, which is immense because it takes about 10 years for a drug to be discovered, to get from those initial stages and get to the actual patient. And a 10 times increase in terms of speed is hugely significant. And pharmaceutical industry has really recognized this, and sticking to the rule of tens has actually increased funding in this area. And so the predictions over the next 10 years are that the actual value of the AI pharmaceutical industry will increase tenfold to greater than $10 billion US. So there is an immense promise in this area, and that's what's led me to pursue my Clinician Scientist Award, working with the GSK, which is a major global pharmaceutical company and has really invested heavily in AI and drug discovery with machine learning and AI tools to rapidly improve the drug discovery process so that we can overcome therapeutics innovation hurdles that have been a barrier to diseases like acute pancreatitis for so long.
Brilliant. Thank you very much, Rishi. Neil, what one thing are you most excited about for the future?
So I think that it's very difficult to point at any one particular technology or therapeutic or disease application because obviously they're all incredibly worthwhile. If we can save people's lives and improve people's quality of life, that's obviously the goal of a lot of things that we do. I think that one of the things I'm particularly excited about and obviously interested and involved in is we know that various disciplines within science obviously interconnected. But I think that one thing that people are doing a lot better these days and why I'm quite excited for the future is that to develop a complex medicine, even something like an LNP with a nucleic acid in it, it needs an incredible breadth of expertise. So obviously you need clinicians, you need material scientists, pharmacologists, immunologists, so on and so forth. And that's something that's not particularly new. But I think that what's happening now is that there is greater support within definitely industry, but obviously within academic institutions as well, like Liverpool, where they're facilitating and supporting those kind of collaborative and interdisciplinary efforts rather than it being siloed. So instead of having a group of chemists working next to a group of pharmacologists and so on and so forth, and there's a lot more support with the university to develop that infrastructure so people are actually not just working alongside each other, they're actually working truly in collaboration and trying to understand and be able to speak each other's language, as it were. So I think that that's something that's really important, and I think that what's evidenced really well of that at Liverpool is the Centre of Excellence for Long-Acting Therapeutics, so CELT, which is the first and at the moment the only one of its kind globally to develop long-acting therapeutics for a number of different applications, not just infectious disease. And I think that seeing the university put so much effort and support behind something like that is a really good example of therapeutics innovation. It comes back to what I was saying previously, that obviously development of new molecules or new types of therapy, whatever they might be, different modalities, is obviously critical to therapeutics innovation. But just as important is taking drugs that are already relatively efficacious and trying to improve that, either to improve their safety, improve their efficacy, however we might do that, and also improve or try and reduce the burden, I should say, of how those conditions might affect the patient's life. So I think obviously that collaboration and interdisciplinarity is critical to any development, but I think that what's quite exciting is now increasingly seeing academic institutions support that kind of thing and help develop those kind of centres of excellence and projects.
So what about yourself, Danielle? So what one thing are you most excited about for the future?
Yeah, so I think with the development of lots of new new types of therapeutics, we have to supplement that with the development of lots of new assays, so experiments, to be able to test these therapeutics and be able to predict how they all react when they're in humans. And so I'm really, really excited for how all these new therapeutics coming in, we're able to kind of develop our experiments to kind of test these and make them specific to that specific therapeutic. So these therapeutics traditionally were administered orally, but as these new types of therapeutics come in, they're delivered in lots of different ways. So you can think about things being injected. So we've got vaccines and stuff like that. So the LNPs that were in the COVID vaccine, they were administered through an injection intramuscularly. And so when we're administering these things, we need to think about how we're going to test them before they go into humans. And so kind of better developing these assays to be able to predict what they're going to be like in humans, and also to better kind of predict the correlation. So we want to make sure that we're trying to predicting how they're going to react in humans in the lab, in kind of like experiments in kind of a dish and stuff like that. So that's what I'm really interested in.
Brilliant. Thank you very much. So there's a lot of great work happening, and the University of Liverpool in this research area has to constantly innovate and move at speed. What do you think we need to be doing more of in this area?
So I think it's firstly really important, considering the sort of nature of clinical conditions that we're increasingly facing with an aging population and also a very diverse population that we have greater access to worldwide, to embrace precision medicine strategies. That whole traditional approach of a one-size-fits-all method isn't going to work in the future, and we've realized that it's not the optimal strategy with regards to therapeutics. So we need to better understand disease heterogeneity for a range of conditions and really get to the nitty-gritty, the background of the biology through hypothesis-driven research and not just technological investment, to understand which targets are best in which people, and to have improved targeted therapies in a precision medicine approach as a result of that. Alongside that, we need to make the technologies to really identify those biomarkers that are necessary for that approach and the drugs more affordable to all. And the things that will facilitate that are incentivizing open sharing platforms and data distribution, collaboration worldwide, which has started obviously as a result of COVID, but we need to maintain the momentum really that was gathered as a result of that to really allow translation to all areas. So I think that those things are key. And alongside that, if we can actually understand conditions better and have better resolution with regards to a variety of diseases, we'll be able to target better treatments to them in the future. And that's certainly the hope I have from the condition that I predominantly treat, as well as a range of conditions that cause critical illness in patients day to day.
Brilliant. Thank you very much. Neil?
So I think for me, it would be a slight extension of what I was mentioning earlier about interdisciplinarity and collaboration. It touches on some of the things that Rishi has said as well. I think we need to be able to put the right people in the room together when you develop these kind of complex medicines. We know traditionally, I mentioned, material scientists, chemists, pharmacologists, clinicians, but we increasingly realise that as these therapeutics become more complex, their interactions with living systems, we need to get much better resolution, greater granularity on how they actually interface with these kind of things. So pulling in expertise from people from disciplines we might not necessarily traditionally think of in therapeutics innovation, say biophysicists, so we can actually look at the actual materials, biology interface, and collaborations around that space, I think is absolutely critical. But I think that one thing that in particular that we're involved in, and we've done a lot of over the past sort of 15, 20 years, is standardization and harmonization of approaches to developing those medicines. So obviously there are international standards and clinical standards and things that we need to develop those materials going through. But the problem is, as these therapeutics become more and more complex, we know that there's a significant reproducibility crisis in science in general anyway, and that can be down to quite a few different factors. But what were things we know that when we look at how these complex medicines are made, how they interact with living systems, we know that to some extent the devil is in the details, and very small, potentially initially insignificant differences or minor differences, I should correct myself and say minor differences in these kind of materials could be what underpins some of the big differences that we see. So we've worked in lots of and led lots of national and international consortia where we've worked with EMA, FDA, MHRA around standardising and harmonising our approaches. So Danielle talked about developing new assays and new models. That's great. The innovation is brilliant. The biology is really interesting. If we want that to be adopted by the community, then we have to look at how can we standardise it, what needs to be done. And a big part of that is metrology, which is something that is, and I'll say I'm guilty of this, I can find a little bit dry myself. Sort of as an immunologist, you want all these sort of really interesting things to happen, but metrology is incredibly important to all of these things, to try and understand whether those slight differences we see in materials are really important, or whether or not they're things that we don't necessarily need to worry about, which is just as important, that has arguably a larger impact financially on the health economic side of things. If we don't need to worry about something, then theoretically we don't have to pay to do it. So standardization and harmonization definitely in our approaches, and that has to be on an international level. But to do that, we need reference materials. So the development of reference materials for these relatively simple, complex medicines, I know that sounds like an oxymoron, but simple complex medicines like an LNP with an mRNA, even that needs sort of reference materials from both the manufacturing, preclinical development, clinical development. And that's something that doesn't really exist for a lot of these materials at the moment, but it's something that we are trying to do a lot more of, something we're trying to push forward a lot more. We have a fantastic collaboration with the National Measurement Laboratory, which is part of the laboratory of the government chemists. So they're involved in lots of international metrology efforts, and I have a role there as academic in residence advising on some of the biological aspects there. So I think that as well as that increased collaboration, we do need tools, procedures, materials that we can use to make sure that everyone is actually pulling in the same direction.
Brilliant. Thank you very much. So, Danielle, what do you think we need to be doing more of in this area?
Yes, so I think Neil and Rishi both kind of pointed out the really key things that we need to do. So collaboration, being able to better kind of work with other people and get the data from other people to be able to develop these therapeutics and to work together to kind of really drive everything forward. And again, as Neil was saying, the models that we need to use they're really crucial to be able to develop these therapeutics. Without them, we can't move those therapeutics into humans and then be able to kind of work to curing diseases or being able to kind of prevent infectious diseases or stuff like that. So I think, really crucial is working together with groups. I work currently on another project as well, so I'm part-time on both projects, where I work with the Intracellular Drug Delivery Centre, and we've been able to kind of form a postdoc group where all the collaborators, there's a number of postdocs working on the project, and we're able to kind of meet up every month, and we kind of talk about what we're working on and how we can help each other to better develop our kind of key areas in the project. So some people work on doing in vitro work, some people work on in vivo work, and being able to kind of help each other out with our kind of experiments and say, oh, I think this might work better because this is what we've seen, is really crucial. Because if we're working on these things separately, we kind of might miss out, or we might think, oh, we haven't thought of that. And you're able to kind of collaborate and really kind of drive forward these tests that we can use to develop these therapeutic innovations.
Excellent. So earlier on we mentioned Catapult. Neil, can you just give us a brief overview of what Catapult is?
So the Catapult Network is a UK government-funded network of academic industry collaborations around different areas of challenges. So we work with Medicines Discovery Catapult, and they are organisations that can support the development, in that particular case, of new therapeutics, new medicines from anyone from either from the actual academic background or working with pharma.
Brilliant. Excellent. Thank you. Thank you very much, everyone, for joining me. Rishi, Neil, Danielle, wonderful to meet you all, and thank you for telling us about your important research. We look forward to following it in the future. That brings us to the end of this episode of Original Ideas, and a topic that's always fascinating, looking at the world of medicine and therapeutics, research and innovation. A huge thanks to Professor Neil Liptrott, Surgeon Scientist Rajarshi, Mukherjee, and Dr Danielle Brain for joining us to talk through their work. If you've enjoyed listening, please do check out some of the other episodes from the series. We've looked at topics such as sustainable business.
It's looking at what responsible business means, not just doing less harm, but how we can actually do positive, restorative, economical impact.
The future of AI.
We really see the AI tools working side by side with the humans who are going to be deploying them.
And what academic research can tell us about popular music.
As an audiovisual media scholar, I'm really drawn to her music videos as another way to capture a lot of different fan bases.
Head to liverpool.ac.uk/research, where you can also sign up to our monthly research newsletter with updates on events, opportunities to work with us, and much more. We invite you to follow and rate the Original Ideas podcast so you can get the latest episodes as soon as they're released. Thanks for listening. Talk to you again soon.